Today, we're going to examine a profound shift happening in medicine, the application of machine learning to the immense challenges of preclinical drug discovery. We'll break down how these computational tools are being used at every step, from identifying a potential new drug all the way to verifying its safety.
So to really understand the significance of this shift, you first have to grasp the scale of the problem. Bringing a single new drug to market comes with an estimated price tag of over $2.5 billion. It's a staggering investment, and it creates an enormous financial barrier to innovation.
And the challenge isn't just financial. On average, the whole process takes somewhere between 12 and 15 years of sustained effort. That's more than a decade, from an initial concept to a medicine that can actually reach patients.
Now, when you combine that cost and that time with this number, the core challenge really comes into focus. Over 90% of drug candidates that enter phase 1 clinical trials are ultimately going to fail. They fail either due to a lack of efficacy or because of some unforeseen toxicity. This incredibly high rate of attrition is the constant battle that researchers and pharmaceutical companies face.
So where does machine learning fit into all of this? Well, at its core, machine learning uses statistical algorithms to find patterns in the massive, complex datasets that define modern biology and chemistry. These models learn from the data to make predictions, and their performance generally improves as the quantity and quality of that data increases.
We've just covered the core problem. So now we're going to walk through the 3 key preclinical steps where ML is making an impact. First, identifying those initial hits, then understanding their mechanisms, and finally, preparing them for translation to the clinic. We'll wrap up with a look at the outlook and the challenges that still lie ahead for the field.
All right, so the very first step is about finding a starting point. It's about finding a hit compound, something that shows some initial promise against a biological target. You can think of it as searching for a needle in a vast chemical haystack.
The traditional way of doing this, shown on the left, is called high-throughput screening, or HTS. This involves physically testing hundreds of thousands, sometimes millions, of compounds one by one in a lab. As you can imagine, it's slow and very expensive. On the right, you see the new approach, ML-guided virtual screening. Here, computers evaluate these chemical libraries in silico, that is, computationally, which allows scientists to search through billions of compounds in just a fraction of the time.
Let's look at 2 key techniques here. The first is the Directed Message Passing Neural Network, or DMPNN. This type of model essentially learns the language of molecular structure. By training on known activity data, it can then predict which new molecules are likely to be effective. This exact approach led to the discovery of halicin, a novel antibiotic. The second technique, deep docking, uses deep learning to predict how well a molecule will bind to a target protein. This was instrumental in finding new inhibitors for a key SARS-CoV-2 protease.
Now, virtual screening is powerful, but it's still limited to searching through molecules that already exist. The next frontier is what's called molecular generation. This is where machine learning models don't just search, they create. They're designed to dream up entirely new molecules, exploring a chemical space estimated at over 10 to the 60th power. That's an astronomical number that just dwarfs all existing chemical libraries combined.
3 major classes of these generative models are really at the forefront. First, you have variational autoencoders, or VAEs. These learn a sort of compressed representation of molecules and can then generate new ones that are optimized for certain properties. This was done with the Jaeger model for antimalarials. Second, there are diffusion models, which build a molecule step by step, essentially by refining a noisy, random input. And finally, you have chemical language models, which treat molecules as a language, using architectures like transformers to generate new molecular structures.
This quote right here shows the real-world potential of these methods. It comes from a study that used a chemical language model with transfer learning. After being trained on a general dataset, the model was fine-tuned on just 46 known inhibitors for a particular cancer target. It then generated over 1 million novel molecules. And here's the crucial part. When a selection of these were actually synthesized and tested in the lab, they showed potent activity.
Okay, so once a hit is identified, the next critical question is how does it work? And this brings us to step 2, elucidating the mechanism of action. To optimize a drug, you have to understand its biological function at a molecular level, and that often begins with predicting its structure.
2 main approaches have emerged for predicting a protein's 3D shape just from its amino acid sequence. On the left, you have models like AlphaFold 2. They rely on multiple sequence alignments, or MSAs, which involves comparing a protein sequence to its evolutionary relatives to infer its structure. This is highly accurate, but it requires that those relatives exist in our databases.
On the right are models like ESM-Fold, which use protein language models. They learn the fundamental patterns of protein structure from millions of sequences, which not only makes them much faster, but allows them to predict structures for so-called orphan proteins that don't have any known relatives.
And diffusion models appear again here, which just shows how versatile they are. DiffDock uses that diffusion process to solve the molecular docking problem, that is, predicting the precise orientation of a drug when it binds to a protein. Taking it a step further, RF diffusion uses a similar principle not just to predict, but to design entirely new proteins from the ground up based on desired structural features.
You know, we can go beyond just single proteins and start to model entire cellular systems. Geneformer is a prime example of this. It's a large transformer model that was pre-trained on nearly 30 million single-cell gene expression profiles. Through transfer learning, it can be fine-tuned on smaller, disease-specific datasets to predict how gene networks will respond to a drug. This helps to pinpoint the precise molecular target of a new compound.
All right, this brings us to the final preclinical stage, which involves optimizing a promising lead compound for potential use in humans. This is where its drug-like properties are investigated and its safety profile is predicted. And this gets to a central question in drug development. How can we get better at predicting a drug's safety and efficacy before it goes into clinical trials? The process involves predicting a suite of properties known by the acronym ADMET. That's absorption, distribution, metabolism, excretion, and toxicity.
This slide gives you a snapshot of how machine learning models are being tailored to predict these critical properties. For solubility, a key factor in drug absorption, one model was able to reduce prediction error rates by 20 to 30%. For cardiotoxicity, a graph neural network proved superior at predicting blockage of a crucial ion channel. And for overall toxicity, a multitask network was actually able to identify the specific molecular fragments, or toxicophores, that were responsible for adverse effects.
So, we've seen how ML is applied at each stage. Now let's take a step back and look at the outlook and the challenges for integrating these tools into a seamless discovery pipeline.
4 major challenges really stand out. First is data. These models require high-quality, standardized data, which means we need a collaborative effort to build better repositories. Second is interpretability. We need to understand why a model makes a certain prediction to trust it, a concept often called explainability. Third is validation. Any prediction made on a computer must always be confirmed with rigorous, real-world lab experiments. And finally, there's collaboration. True progress here requires breaking down the silos between computational scientists and experimental biologists.
This brings us to a final thought. The integration of machine learning isn't just about making drug discovery faster or cheaper. It's fundamentally changing the questions that can be asked in the first place. So, as these computational tools merge more and more with experimental science, we have to consider how they will reshape not just a single process, but the very nature of biological investigation itself.